Hi, Dr. Amos. Here, this is my engagement table. This is take two because my other one got locked up during publishing, and so it just deleted. So the first standard I hit was MSLS 1-2. Develop and use a model to describe the function of a cell as a whole in ways parts of cells contribute to the function. So for this standard, the students were learning the roles cells play in living organisms, and also the roles that the organelles play in the cell, the individual organelles, and how they contribute to the job of the cell. So this was a deep standard that we spent several days on, and they learned using peer teaching, teacher-led, and individual research to get their understanding down. And to prove that they learned, we had them, as in we, my partner teacher and I both did this in our classrooms, had the students create a poster that related the organelles of the cells to something in their everyday life. And so they first had to choose whether they were doing a plant or animal cell, and then they had to put all of their organelles in there in the poster, and half the poster could be printed images, but at least 50% had to be hand drawn. And so so they'd have to have like a picture of the Golgi apparatus, what it looks like in a cell, and a picture of what they relate it to in their everyday life. One thing that I relate the Golgi apparatus to is a mailman, because they package and distribute proteins throughout the cell. Mailman packages and distributes packages to people, and so that was kind of how they did it. And then to completely assess their understanding and to make them demonstrate their learning, they had to come back, and we had an individual meeting with them that took a couple minutes, and they grabbed 5 popsicle sticks of the organelles with different organelles on them, and they had to explain their functions, and they could use their poster to help them explain the functions, because the poster only had the name of the organelle and the pictures. So if they were able to make that relationship, they were usually able to understand what that organelle does, because they look at the poster and say, oh, I have a mailman. Yes, because they package and distribute. And they were able to retain that more. And so for the types of how engaged the students were, the students were pretty engaged doing this because they got to make it their own. They got to be creative, like Hattie says, using creativity to help them learn. And they also provided opportunities for students to talk about themselves, because when I was like going around when they're making their posters, I would ask them like, oh, that's interesting. Why do you think that relates to, or how do you make that connection? And they were able to go into this big elaborate thing of how they made this connection. And that's all that I needed to know, because they were able to understand it. The students are very engaged. They were able to make it their own. And it was to their artistic ability, and also like their effort. Like, not all students are gonna prove a master art piece of a poster, but as long as they're able to make the connections, that was what we were going for, and it proved to be very successful. My second standard was MSLS 3-2, develop and use a model to describe why asexual reproduction results in offspring with identical genetic information, and sexual reproduction results in offspring with genetic variation. For this standard, the students were learning how asexual reproduction is one parent and they produce identical offspring, and sexual reproduction is with 2 parents, and the offspring has a combination of each parent's genes. And this again was taught over a whole genetics unit. And we did a recipe for traits activity to assess their understandings of the topic, and they randomly had to draw 8 genes out of an envelope in whatever order they got it. So they all had the same 8 genes, but different order meant different. They'd get a different end result, because gene 1 there's 4 different like head types for gene 1. And so they would get a different head, and then gene 2, different body, and so on and so forth. And so the order, just altering 1, changing 2 around, caused the dog to look different. That's how you get your genetic variation. So they'd make their long strip of genetic code, And then they'd have to draw their dog based on the traits that match up their genetic code. And as a result, the students were able to fully understand the difference in asexual and sexual reproduction. And they created the model, they created the genetic code, they drew their dog to show the traits that it would have had, and were able to make the connection that the dogs reproduce sexually because of genetic variation that they had compared to other students in their class, or the other students in their class, and their other students in their grade. I have 125 students, and none of them looked identical or had the identical same exact genetic code. And it was only 8, so that was able to blow their minds because we have so many more genes than 8. And so they were able to really grasp that. And again, for this one, I used creativity that Hattie talks about on page 44, and also high energy as a stimulus for engagement, because they were engaged because they got to make it their own, and it's dogs, and they all loved dogs, and they were all excited to create the best dog. And then some of them were like, I got all the worst traits, I got a big body and little legs or whatever and stuff like that. But they were excited to try to also match it with different species of real dogs. And so that was exciting. They were engaged, and they were able to answer. Following this, they had to answer questions about asexual and sexual reproduction and the differences and the results of. And again, as a whole, my class did very well with this because they were able to grasp it. And they were engaged the whole time. So my 3 and 4, standards 3 and 4, the activities I did were super engaging. The students loved them. These were definitely my best. For standard 3 was MSLS 3-1, develop and use a model to describe why structural changes to genes, mutations located on chromosomes may affect protein and may result in harmful, beneficial, or neutral effects to the structure and function of an organism. So this was also big. I had to teach my students, my 7th graders, what neutral meant. That was a learning curve. I just assumed that they knew what neutral means, and I know in a 7th grade classroom, or in the classroom, you never assume a student's knowledge. Lesson learned. So we talked about what it means to have beneficial, harmful, and neutral traits, or something that is beneficial, something that is harmful, and something that is neutral. And the students spent several days learning about mutations and their effects on organisms, and how mutations are not always harmful. Mutations get this bad rep that they're always harmful, and oftentimes mutations can be beneficial. Other times they're just neutral. So we did this alien activity, and they were in small groups of 4, and they each had to draw a genetic code from the bag, and then look at the key, and it'd tell them what mutation they got, or if they got the normal. So for there's 3 different activities we did. There was locomotion traits, locomotion, eyesight, and finger dexterity. And so we ran the test 3 times, and we recorded our results on the table. And so, for instance, they had to get their alien species each colored Fruit Loops. And so for color eyesight, the normal was blue, so they had to look through a blue lens. One mutation was looking through a clear lens. One mutation was purple lens, and one mutation was a red lens. Obviously, the clear lens mutation would be beneficial because when it comes to sorting the colors of the fruit, because it had to sort the colors, that was the activity that had to sort it looking through whatever color lens. Well, clear, they were able to sort it in 6 seconds, whereas red was a harmful one because it was nearly impossible to decipher the warm color, the yellow, orange, and red Fruit Loops from each other when looking through a red lens. They are so similar that it was really hard, and so it took them 2 minutes sometimes to get them sorted because it took several tries. And the kids loved it because they're competitive. It brought in that mild competition as a stimuli, and also the inconsequential competition because they weren't out anything. They still got their Fruit Loops, but they were able to see how mutations were beneficial, mutations were harmful, mutations were neutral. And so when we finished the each activity, they asked, okay, which mutation was beneficial for this one, which one was neutral, and which one was harmful, and they had to answer that. And then following this, they also use physical movement because, like, the locomotion gene, they had to walk to get their food a different way, whether it's on their tiptoes, normal walking, shuffle walking, or walking on their heels. And so they were up and active and working as a group, and they were competing with each other, but it wasn't like rough competition. It was super engaging. The students loved it, and the best part was they got to eat Fruit Loops afterwards. They loved that part the best, I think. But they also showed a great understanding of that standard. The next standard I want to talk about is MSLS 4-4, conduct an explanation based on evidence that describes how genetic variations of traits in a population increase some individuals' probability of surviving and reproducing in a specific environment. So in this lesson, students learned about natural selection and the idea that if a trait is beneficial, the organism with the more beneficial trait will be more likely to survive longer and able to reproduce more and have more successful offspring. So the activity that I wanted to kind of spotlight this on was called Battle the Beaks. So after we spent some days learning about Darwin's research on the Galapagos Islands and his findings about the different birds on different islands and the food they eat and all this stuff, the students worked again in small groups of 4, and they each got a different beak. There was binder clips, clothespin, fork, and tweezers, and to see which was the most successful with each type of food, and then which was the most successful overall. So there's 4 different types of food. There's clothespins, rubber band, or 2 different types of foods. I'm sorry, pinto beans, macaroni, rubber bands, and paper clips. And so first we did a round with just one type of food, so they would compete to pick up the most of pinto beans, just pinto beans, and then we recorded that in the results in the table. Usually they could only eat one at a time, so the fork was at a disadvantage. The tweezers usually won. And then the macaroni, again, the tweezers usually won. The fork did pretty good, but the clothespin and the binder clip mostly broke the macaroni. That caused a huge mess. I mean, when we were doing this activity, there was macaroni flying pieces of macaroni shattering and flying everywhere. And beans also, because of especially the binder clip, the pressure, they could not. The binder clip students struggled, but so this brought in physical movement again because they were standing there competing for food in the middle of the table, and it also brought in that inconsequential competition because they were competing for food, but it wasn't also a fair fight, and they were able to make those connections, and then so as a result, they had to fill out the table for every simulation we did, every activity we did, and then they had to answer questions. And a lot of the questions were like, okay, so we know that, so who got the most food, who got the least food, and what does this mean for our next generation? So then they were able to look at the table, who got the most, who got the least, and then the vast majority as a whole, the students were able to understand that the one that got the most food, that's what the next generation is going to look more like, because they're going to be the ones that are fed. They're the ones that are going to survive better, have a better chance at survival. And so they were able to make those connections again. And this was super engaging because they weren't just sitting there. Their minds, they were competing. They were seeing it happen in real life. And these are so engaging. It was so fun to interact with the students on these because they just had a blast. Now, my 5th standard, MSLS 4-6, use mathematical representations to support explanations of how natural selection may lead to increases and decreases of specific traits in population over time. Now, for this standard, the students used evidence and the math behind the idea that natural selection results in increases and decreases of traits found in the species. So the students did an online simulation of the peppered moths during the Industrial Revolution in Europe, and the result of the simulation, the students had to answer questions based on the simulation and predict what would happen in different situations. If there was no predators, would the moths change colors? Would the colors of the population of moths change depending, and all this stuff. And this is more of a self-stimulus for engagement. And this